Hello everyone, welcome to Last Night Guru. So in this video we will be discussing about biotechnology principle and processes. So we are going to study the series of the chapter. So today is the first part, that is about introduction of what is biotechnology. So let's get started. So first of all, what is this biotechnology? So as the name suggests, bio in the sense living, right? So technology ಅಂದ್ರೆ ಏನು? So using technology in living organism or enzymes. Why we are using technology in living organism? Because we want some products from that living organism which are useful to humankind. So that is why biotechnology is very much in trend, because so many vaccines, so many products we are taking from out of biotechnology. So let's see the definition. Biotechnology is the technique, as I said, it is a technology using what? Using living organism and also their enzymes. Why? For their products and processes which are useful to human. So there was a federation which was called European Federation of Biotechnology. They gave a specified definition for biotechnology. So let's see what does European Federation of Biotechnology says. So according to EFB, biotechnology means it is a integration in the sense it is the development of natural science, organisms, cells and molecular analogs. Molecular analogs involves DNA and RNA. So development of all these things. Why? For products and services. So this is the specified definition for biotechnology given by EFB. So now let's see where all this biotechnology is useful. So biotechnology primarily it deals with microbes and mediated processes. Microbes in day-to-day activities we can see making of curd, bread, wine, etc. But in large scale we need the help of biotechnology to make this curd, bread, wine, etc. There we can use biotechnology. Next is in vitro fertilization. In vitro fertilization in the sense fertilization, fusion of the gametes inside the lab. So that is in vitro fertilization, which we also can call it as test tube baby. So this is also a technique of biotechnology. Next, synthesis and using of gene. This is very important. Mainly biotechnology plays a very important role in this process, where a gene can be modified and various products can be extracted from that gene. I hope you all know what is gene. What is gene? Gene is a segment of DNA which codes for a particular character. For example, tallness is a character. So this character is acquired from a specific gene. Okay, so for example, this is a DNA. So this is one gene which codes for height, and this gene produces a characteristic for height. Okay, so next, preparation of vaccine. Various vaccine is produced from the DNA. Even one good example which is a product of biotechnology is insulin. So next is correcting of a defective gene. Even now biotechnology is widely used to correct the genetic disorder by correcting the gene. So all this area biotechnology plays a very important role. Next, let's move to the principle of biotechnology. So there are two main principles of biotechnology. The first principle is genetic engineering. So genetic engineering plays a very important role in biotechnology. Most of the process of biotechnology involves this genetic engineering. Let's see what is this genetic engineering. Genetic engineering means gene. Okay, you're engineering the gene means you're altering the gene. Why? To obtain some products. Let's see the definition. The technique in which the genetic material, which is the genetic material, the DNA or RNA, is chemically altered. What we are doing? We are altering the DNA. For example, this is a DNA. Okay, So inside this DNA, I will insert a gene which codes for insulin. What I'll do to this DNA, I will add a gene which codes for insulin. So then what I'll do, I will insert this gene into a host, any host. It may be plants, it may be animal, or it may be most commonly used host is bacteria. So I will insert this into the host. So this this host carries a gene for insulin as well. It carries its DNA. Okay, now can it start produce insulin? Yes, because its DNA has insulin coding gene. So this insulin then I will extract and use to produce insulin hormone. Okay, so let's see the definition. The technique in which the DNA or RNA is chemically altered. See, I altered it by adding a insulin gene, and then what I did, and I introduced it into a host organism to change the phenotype. Phenotype means, what is phenotype? Its character. Okay, so did it change its character? It used to not produce insulin, now it started producing the insulin. This is the change in the characteristic. So this is the first principle of biotechnology. Now the second principle is bioprocess engineering. This is also very important in biotechnology. What is this bioprocess engineering? See, for all this process to take place, there should be a very clean and sterile place. The place should be very clean and sterile and very hygienic. So maintenance of sterile condition is very necessary. Why it is very necessary? Because see, I am cultivating this host organism. I am cultivating this host organism into a culture media. So if I don't maintain a sterile environment, if I expose this into an external environment, there are many bacteria outside, because bacteria are cosmopolitan, they are everywhere. So I may get unwanted colonies here rather than my interested host. So that is why maintenance of sterile condition, why? To get our desired host is very necessary. Let's see what is this bioprocess engineering. Maintenance of sterile ambience in chemical engineering process for, as I said, growing of desired microbe. If I want to grow only this microbe, I have to maintain a very clean and sterile area. Okay, a desired microbe or eukaryotic cell in large quantities. Why? For the manufacture of antibiotic, vaccine, enzymes, etc. So this is the second principle of biotechnology. So now let's discuss about RDNA technology. So RDNA means recombinant DNA technology. So the whole chapter is depend on this recombinant DNA technology, and this is very important process of biotechnology. For this the process, we should know what is RDNA or what is recombinant DNA. So I will give you a simple example. What is this RDNA? See, if suppose now I want to synthesize lots of insulin. Okay, you all know insulin, that injection will come in market, no? So I want to synthesize lots of insulin. What I'll do? So before some years, insulin was obtained from pig pancreas, but people start developing allergies from pig pancreas because of incompatibility. So then some companies came with RDNA technology to synthesize this insulin. So let's see how insulin has been taken using RDNA technology. So first I will isolate some DNA or from the human, which consists of this gene of interest, that is insulin. For example, this is a DNA of a human, and here is our gene of interest, that is the gene which codes for insulin. So I don't want all this gene. I am only concentrating on my gene of interest, which is that is insulin gene. Okay, so what I'll do, I will isolate this DNA from human DNA, human cell, and then I will cut my gene of interest. So now I have my gene of interest. Okay, that is insulin coding gene. So now, to get lots of insulin, I have to put this into a host organism. Which is the host? Most commonly the host will be bacteria. But one problem is that I can't insert directly my gene of interest into the host. Okay, so it needs a vehicle or a vector. So here plasmid, which is plasmid is a circular DNA which is present in the bacteria. So this circular DNA, that is plasmid, acts as a vector. So now I will insert my gene of interest into the plasmid or the vector. Okay, so I have inserted my gene of interest in this. Now I can call it as RDNA or recombinant DNA. Okay, after forming this recombinant DNA, this is the recombinant DNA is transferred to the host organism. So now the host organism, because it has the gene of interest, it will start synthesizing insulin, and this host can be synthesized in a large scale to get a large amount of insulin. This is the principle behind RDNA technology, and the whole process depends on this. So let's see which are the steps involved in this RDNA technology. So first step is isolation of the genetic material. As I said, first what you will do, you will isolate the genetic material, or you will isolate the gene of interest. Now next what you will do, you will cut your gene of interest, isn't it? So that is the second step, cutting of DNA at specific location. Now after cutting your gene of interest, you will get only one gene of interest, so you have to make its more copies, so that if one is lost, you have many copies. So the third step is amplification. Amplification of DNA means you are making many copies. How you are making many copies? Using PCR in the sense polymerase chain reaction. Now what you will do, you will insert the rDNA into the host organism. Before that, you will insert into the vector, and then you will insert into the host cell. Next, you will obtain the product, that is, here you can see you obtain the product. Now downstream processing means the product has been packaged and quality is being checked. So all this is involved in downstream processing. So in this whole chapter, you are going to study about these steps of creating RDNA. These are not just 6 steps. So this involves lot many processes. So you are going to study one by one. So in the next class, we are going with the first step, that is isolation of the DNA. So don't miss it. Keep watching. Thank you.